my pleasure and honor to introduce tonight's speaker, Mike Schaadt. Mike has been director of the Cabrillo Marine Aquarium for almost 10 years. He earned his bachelor's degree and his master's degree in marine biology from Cal State Long Beach. And he's been involved in formal and informal marine science education for more than 35 years. He has authored over a dozen journal publications, book chapters, and 3 books. He is very good at finding novel ways to motivate people to learn about and care about the oceans. His research interest is the natural history of plankton with special emphasis on deep sea forms. And at this point, I would normally razz the speaker, but since he's my boss, I will not. The title of tonight's talk is The Vast Frontier, Tales from the Darkside. Please help me welcome Mike Schaadt.
No, I'm sorry I started all that. It really is a pleasure to be here. I always tell you that when I get a chance to come up here and introduce a speaker, and now being the speaker myself, this really is a pleasure for me. Before I go any farther, though, I have a special guest here tonight. My daughter, Erin Oderlin, is in the audience. Erin, a water biologist with the City of Los Angeles, a third-generation City of Los Angeles worker over at Hyperion Wastewater Treatment Plant. So it's a pleasure having Erin here tonight, and she gets a little extra star because she's had to live with me and this for all of her life, and she still comes in and listens to it. So what we're going to be talking tonight is something that I certainly enjoy, and so for me this was an easy thing to choose. So I'm excited about it. This is that vast frontier, and thanks to Gene Roddenberry. It's one of those communicators. I was wondering how many people I had to explain that to. But really, I'm going to be talking about Tales from the Darkside. So this really, thanks to Gene Roddenberry and George Lucas, that's where these titles came from. And actually, The Vast Frontier is the name of an exhibit that you'll be seeing pictures of here in just a moment. First of all, just the facts. By the way, those of you that have been kind of groupies of our lecture series, you may remember that Dr. Shauna Gofretti from Occidental College visited us a little over a year ago, and she talked about the deep sea. So I actually made it a point to go listen to and watch her talk again the last week or so to make sure I wasn't doing too much overlap, because she certainly talked about the deep sea. And just as a little bit of a teaser, we have Dr. Ann Boll that's going to be coming and talking to us in just a couple months, and she's going to be talking about the deep sea too. So you're going to get a good sampling of the deep sea. But I want to give it from more of an overview, and I want to give you what I refer to as a rogues gallery of the kinds of animals that you find in the deep sea. And then there's a couple stories that I want to share, I think will illustrate just what a strange and exciting world this is that we don't know much about. First of all, not surprising to any of you out in the audience, I'll bet, the ocean covers over 70% of our planet. People talk about quite a bit from space. This would have been called better ocean planet rather than Earth planet. It provides 99% of the living space on the planet. I want to let that sink in for a minute. Because I think when all of us think of living spaces, even myself, besides thinking of the furniture store, I think of where things live, and I think of the land, and I think of trees, and I think of burrows, and I think of things like that. And yet, because it covers so much of the planet, and because there's a third dimension that has that depth to it, it really is a huge living space. And that's really where we need to be spending a lot of time to get to know it. It provides more than, or about 50% of the oxygen we breathe. So everybody, do this with me. One deep breath, ready? Okay, let it out. Okay, now another one. Thank you, ocean. That's what we describe it to the children that visit us, is that every second breath you can thank the ocean, because the plants that are in the ocean are providing 50% of the oxygen that we breathe. So it's pretty important. And even more important nowadays with climate change, It absorbs 25% of the carbon dioxide that we are producing as humans when we burn fossil fuels. This is an important point that I'm going to bring up at the end of my talk, but I just want you to remember that. This is one of those typical National Geographic pictures that you see, and the pink kind of area that you see, it's kind of hard to tell because it's a topographical map, showing you the land, and then the yellowish is showing you the ocean bottom. So I wanted to point out that over 2.5 miles on the average is if the whole world, the whole Earth was covered just by the ocean, it would an average of 2.5 miles. That hopefully is starting to sink in for that 99% of the living space, because unlike the air that supports some animals and plants, in the ocean, the entire space from the bottom all the way to the surface, including the surface, is inhabited by animals in one way or another. This is what you see when you look out from Palos Verdes and you look out across the ocean when you're on a boat. And yet, what I wanted to point out, I think is illustrated pretty good in this illustration here, is somebody saying, day 44, still stranded, with nothing but flat, empty water as far as the eye can see, missing all the activity going on underneath the ocean for an average of 2.7 miles. Some of the terms I'm going to be using tonight, no, you do not have to remember these. Those of you that are students, you don't even have to write them down if you don't want to. But I'm going to be talking about this offshore area. It's referred to as the pelagic zone, the oceanic zone. When it's close, there's the continental shelf, which here on the west coast is pretty close to our land, whereas on the east coast it goes out hundreds of miles and you're still not in very deep water. Here you get into deep water pretty quickly, and when you go out, it's anything offshore is called pelagic. If it's close to the shore, it's called neritic. And then if it's off that continental shelf, it's referred to as oceanic. Then that third dimension from top to bottom, you have a zone that is kind of described by light being able to penetrate from the sun. And that's called the photic zone. And then you have the aphotic zone, which is below that area where light actually penetrates. And that's the vast majority of living space actually is dark all the time. Eternal darkness. Except for bioluminescence, which we're going to talk about tonight. So I work for an aquarium, and I work for this aquarium, so I figured, why don't I just take pictures of our own exhibits when I talk about these things? Because we have a whole exhibit space that talks about the open ocean. However, I've got to apologize that I took these pictures, not our photographer, Gary Florin, who would do a much better job. But for this particular part of the talk, it goes pretty quick, and I think you'll get the gist of it. So the very top, and I want you all to follow for just a minute. Everybody make a cake. So you're holding on to your cake just like this. Everybody do that, please. In the spirit of John O'Geen, do it, do it. Now you have that 3-layer cake. The top is the surface of the water. The bottom is the mud and sand down below. Okay, now I want you to take the very top. It's a 3-layer cake, folks. Take the top layer. You can put your hands down. You did a very good job, by the way. I think I should have taken bets as to how many I could get you to do that. This is called the EPI Pelagic. That top layer of your 3-layer cake is called the EPI Pelagic. That goes from the surface down about 200 meters. So that'll give you an idea. Now the next one is the kind of that mesopelagic zone, that middle zone. So make your cake again. Man, you guys are great. And you've got the top epipelagic, you have the middle mesopelagic. Well, that's what we're talking about now. And we refer to that as the twilight zone because during the day, there's enough light that goes down there that there's light that the animals can see. However, not enough to support plant life. That goes down to about 1,000 meters in the clearest areas of the open ocean. So that's what we refer to as the twilight zone. By the way, when we did this exhibit 20 years ago, we were trying to get kind of people into this idea, the twilight zone. That was Rod Serling's TV show, right? Those of you old enough to know that? Nowadays, that's not the way it's seen. Now it's all about vampires. Still works. And then the final bottom part of our three-layer cake, folks, is where the sun doesn't shine. Ever. Eternal darkness. And the point that I want to make to you is that 200 or 300 years ago, a lot of scientists, a lot of people believed on the planet there was an area that once you got below that area in the ocean, there was no life. We now know that to be far from the truth. There's plenty of life down there, even in the deepest part of the Marianas Trench, the deepest part of the ocean. The animals live on the very bottom. And that goes down from Bathy Pelagic, below 1,000 meters all the way down. And there's other names like Abyssopelagic and Hadopelagic. Don't worry about that. I wanted to talk a little bit about the physical parameters that these organisms must put up with if they live in different depth levels. So the first one I wanted to point out is pressure. Pressure increases as the depth increases. To illustrate that to you, how many of you have gone into a pool before and you've gone down to the drain? Raise your hand, I want to see. Oh, other swimmers, I'm so excited. Well, the point that I think you would all agree with is as you go deeper, it actually starts hurting your ears. And that's because the pressure on the outside is pushing on the tympanic membranes, your eardrums, and it's pushing them in because the pressure on the inside of your head is actually less than the outside. It's pushing on it. And you have to equalize it by holding your nose and breathing out easily. Well, there's animals that live all the way down to the bottom. And one of the things I like to point out in our particular exhibit is we show that with a styrofoam cup. Styrofoam is a type of material that is filled with air passages, air pockets throughout. So once it's been squeezed by pressure, whether it's by your hand or whether it's by the water pressure, it stays to the shape that it was squeezed to. So that's what a cup looks like on the surface. This is a cup that was sent down by some friends of ours on a submersible, and it was squeezed or it was shrunk to that particular level there. This one down here went down over 12,000 feet deep. Same cup. And when it came up, it kept that same shape that it was shrunk to. By the way, I also like to point out that this particular cup on the bottom, the submersible that we sent it down on, and remember it has to be on the outside of the submersible, not the inside where the people are. But on the outside it was shrunk. That cup went down to the Titanic, the wreck of the Titanic. Because we sent it down and some friends that were going on the Mir submersibles, and they were taking pictures for National Geographic, and they took our cup down. So if you ever want to see something that's been to the Titanic, that cup's been to the Titanic. Light also decreases as you go deep into the ocean. So in the middle of the day when the light is coming down, it's coming down, and I know you've taken many of us have done this even as children. You take a prism and you have sun coming through it. What do you see on the other side of the prism? You see a rainbow, a spectrum. That's to tell us all that white light is made up of different wavelengths of light. Well, those wavelengths of light have energy to them, and there's different energies for different wavelengths of light. Red light is taken out in the very surface waters. It's not very energetic. It doesn't go very deep. So what we did, we took color wheels, and the color wheels on the surface looks like this. And then as it goes deeper, you can see that only the more energetic light is showing up. So the blues and the greens and the purples and the violets, those are the ones that show up until you eventually end up with no light whatsoever at any time. So light decreases with increasing pressure or increasing depth. Finally, temperature. As you go deeper in the ocean, temperature gets less. So it could be 60 degrees Fahrenheit on the surface, and there's parts of the ocean where it's actually below freezing in the deepest part of the oceans. But because of the pressure and the salt, it does not freeze the water. So it's below freezing, but it doesn't freeze. Animals live there. Animals have fluid in their body. That fluid doesn't freeze either. Isn't that interesting? And so these fish actually have almost like an antifreeze in their fluid of their bodies that they can live at that great depth and those very low temperatures. Now, I'm going to show you that rogues gallery because to me this is always one of the most fun parts. And I'll have some stories that go along with each one of the animals, and then we'll tie it together right at the end. But I want to be truth in advertising. Open ocean includes a lot of different animals. In fact, all phyla, as we say in biology, all the phyla are represented in the open ocean in some form or another. But we're only going to be talking about the invertebrates tonight, mostly invertebrates. There'll be a couple vertebrates. You won't be learning about big fish. Sorry, Jeff Landesman, if you're here. He's our chief aquarist. You won't be learning about sharks. Yep, I know, you were all excited about that. You won't be learning about seals and sea lions or seabirds, at least not from me. And we won't be learning, sorry Diane, I know, whales and dolphins. This is the realm where you find all of these animals, the open ocean. However, we're going to be talking about the ones that people don't spend quite as much time on. And yet, when it comes down to it, the numbers, the sheer numbers, are so much greater than any of these other animals, by far. One of the ways that we sample them by going out is by taking a boat like the yellowfin from the Southern California Marine Institute. We've been lucky enough to go over the years and be part of a trip where we go out and drag a net that I'll show you in just a moment. You can see that it's 76 feet long, takes out about 40 people. Maybe there's some Long Beach State students out there who have been out on this before. I know I've been out on it many times with Long Beach State programs. When we go out, this is a science trip for us, so we take data. It's not going to do any good if we take a sample of where these animals are from and don't know exactly where they're from, or we don't know what the depth was, we don't know the time of the day, we don't know the time of the year. That makes that sample not near as important to science as if we keep that information. So every time we go out, I have the people that go out with me anyway help me collect that information from the crew so that this becomes important data. We do spend a lot of time on that boat just kind of standing around and talking amongst each other and having a good time, so that's certainly what we do. And then we put down this rather unusual net called an Isaac's Kidd midwater trawl. And because it's long, we always say the IKMT, and many times we even say the IK. John Isaacs, and I can't remember what Kidd's first name was, maybe some of you know, Professor Kidd from Scripps. They designed this net, and how it works is that there's a spreader bar on the top that is hooked up to a cable that comes up onto the boat that goes through this big A-frame and then into a spool, and that's a spool of cable. And that is let out so that the spreader bar goes down. This big metal paravane, as it's called, digs into the ocean, and it's very heavy. So it keeps the bottom of the net dragged down, and the cable keeps the opening of the net open. So as this gets into the water, it's a big open mouth that's being dragged behind the boat. Very slowly, by the way. Very slowly. And by virtue of that, by the way, we don't catch the bigger animals. They get out of the way, no problem. We get those animals that are a bit more lethargic in their swimming, most of them planktonic, which means they're at the mercy of ocean currents. This is what it looks like once the net is deployed. It stays down for about an hour and a half to 2 hours because we want it to go deep, maybe 2,000 or 3,000 feet, trying to get those animals. And there we are, just kind of hanging out on the deck of the boat. While it's there, Todd Chapman, I bet you recognize yourself right there. Yeah, Todd's in the audience tonight. He's now a full-fledged marine biologist, doing the good environmental work that needs to be done. But this is back in your younger days, Todd. And he's deploying a net we call the Newston net. The Newston net is sampling the top about 5 centimeters, 2.5 to 3 inches of water at the very surface. And that's what we're sampling there. And you know what? There's organisms that spend their entire life cycle in that body of water, that part of the water. No other place do you find them but right there in what we refer to as the Newston. This is Todd taking that, why are you laughing? This is Todd taking the sample out, and we're putting it into jars. And this is where the hard sell comes, everybody. Everything's pretty small. So we're all looking at things, and I'm starting to identify things because I've seen them a lot. Todd's helping me out. He's saying, oh yeah, we see that, that's this, this, and this. And other people are shaking their heads saying, they're just making that all up. We do put it under microscopes, though, and we continue our taxonomy, something my daughter knows all about, right? Identifying those animals. That's what you have to be able to do. She's wincing in the eye. I'm going to pay for this, I'm just sure of it. Here's Todd and I working on the cod end, which is what we call the end of that long, big Isaac's Kidd midwater trawl. It ends in this cod end, and when that happens, we eventually unscrew that PVC pipe and then put out the goodies into a container like this, a clear container, and then we start using very high-tech tools to remove the animals. Yes, I'm sure you can see that is a bent plastic spoon. High-tech. Fisher Scientific doesn't get a lot of money from mesopelagic studying people. By the way, we even made them more high-tech. We put holes in the bowl of the bent spoon, so you can literally pick it up, and it's kind of like a colander where the animal will stay in and the water drains out. I'm telling you way too much information. Here we are at night when the net finally comes up, and we try to do this at night because many of these animals diurnally migrate. What that means is during the day, they stay in the dark depths. And then as the twilight continues up, they follow up that darkness. And we believe for most of these animals it's a way to avoid predation because they're harder to see when they're following the shadows, all the way up to the very surface. There's organisms no bigger than your pinky finger that make a migration of 3,000 to 4,000 feet every single day of their lives. In fact, they're literally following that diurnal migration throughout their lives, all day, every day that they live. Must be important to them if they do that. You can see more high-tech pieces of equipment, these clear ladles, and we ladle things out. This is a close-up view, and what I want to point out here is notice the colors that you see, and maybe notice the colors that you don't see. So I'm sure that you would agree that there's a lot of red there. There's black, which is really kind of the absence of color, I suppose. And there's a lot of clear. And those are themes in the bodies of these animals for their coloration and the way that they're designed. Here's one of our participants in this particular class, and she's holding on to one of the largest of all the mesopelagic animals that we get. This is a deep-sea shrimp that we get. And again, notice the colors of it. And notice that it's red. Red in the deep really turns out to be black because there is no light to have the color red come out. Now we're going to start with that rogues gallery. And what I've done for you is I tried to put the phylum here as well as either a class or an order or a family that is kind of a commonly named grouping. So you can kind of get an idea of what they're from. So this one is called Bioluminescent. It's in the Cnidarians. It's a hydrozoan. Bioluminescent is interesting that for years when I was in undergraduate and then grad school, we would go out on trips and find Bioluminescent, and I thought they all looked like this. No tentacles. This is what the Monterey Bay Aquarium found when they put their submersible under and just started taking video of the animals that they saw. Bioluminescent actually has long tentacles. And what we've noticed is when the animal is bumped or somehow threatened, it throws off those tentacles and they're bioluminescent. So imagine if you were a predator of Bioluminescent, you bump up against it because maybe you didn't even know it was there because it's clear, and you're getting ready to bite it, and all of a sudden you have all these little string-like structures just flashing to beat the band. Certainly it's going to distract you, and that's exactly why we think Bioluminescent does that. Another hydrozoan that's found in the deep sea is called Atolla. Atolla, what a great name, huh? Notice the coloration, bioluminescent, by the way. Talking about bioluminescence, this is aquaria. And aquaria is an animal we get every once in a while here at the aquarium. They are also found in the deep sea, but you can find them in the surface waters. And this is what aquaria looks like when it's put under a black light. They actually bioluminesce at the rim of the bell in little packets. And these little packets are filled with green fluorescent protein. In the wild, the bioluminescence takes place inside the tiny packets, and it excites the green fluorescent protein in the outside packet, and it lights up that green color. I'm making a big deal out of this to introduce bioluminescence, but also to let you know that green fluorescent protein, this right here, has been cloned. So it was first identified way back in the 60s, 70s maybe. And once it was identified and collected in large numbers, it was used in a lot of different research, like nerve research, because you can attach it to nerve cells under a microscope, and when you excite the nerve cells without green fluorescent protein, it's an electrical impulse and nothing happens. But if they have green fluorescent protein attached to it, they light up. You can literally follow the pathway of a nerve as it innervates throughout the brain or throughout the muscle that it's innervating. So it's pretty amazing. And if you ever want to know more about this, there's a lot of websites that talk about it. One of them that's my favorite is called Brainbow. And what they do is the green fluorescent protein has now been cloned, and it actually glows different colors. So you can specifically tag certain neurons or other things you're looking for, and it will light up a different color depending on what pathway you're looking for in the brain. So quite exciting in terms of what it does. By the way, the labs that did this pioneering work received the Nobel Prize in Chemistry about 10 years ago. So pretty big stuff. Now this is right out of one of my favorite bioluminescent pages. It's called the Bioluminescence page by Dr. Steve Haddock. Yeah, see how simple that is? From UC Santa Barbara. And Steve and Dr. James Case put this together, and it's all about bioluminescence. Any of you interested in bioluminescence in the ocean, and on land too, by the way, they study that too, you should really jump on this website because it gives you a lot of great information. But I wanted to point out that there's different uses of bioluminescence. Startling, counter-illumination, I'll give you an example of that in a moment. Misdirection, like a smoke screen, another example coming. Distractive body parts, Farnima. Sacrificial tags, burglar alarms. You can see there's just plenty of very simple reasons of why animals bioluminesce. And up to, well, let's see, I think I went through it too quickly on that last slide. More than 80% of ocean animals are bioluminescent. Wow. Wowie. Top that, whale people. So here's another animal you'll find in the deep sea, by the way, and it's related to jellyfish. In fact, you can call it a jellyfish. It's in a different group called siphonophores. And this one is called Ninomiya. Ninomiya is interesting because, like all siphonophores, it has many different parts, like a float that is gas-filled, by the way, with carbon monoxide. This is also the swimming bells. And then as you get down here, there's individual parts that have very specific purposes for the colony. Some scientists believe this is a colony, and some argue that it is an individual. The reason they argue that it's a colony is because those parts are so independent, they can literally live independent for some time. And so there's a lot of not a whole lot of argument, but a lot of discussion amongst the scientists. Another thing I like to point out, Ninomiya is probably, hold up your index finger, everyone. That's about how long Ninomiya is. However, now I want you to hold it and imagine your other finger being all the way down to the floor. That's how long their tentacles can be. So they're this big and they go all the way down to the floor from here and collecting food all the way as they go. Also, with that carbon monoxide float, these animals make a migration every day, a diurnal migration. And when you're on a ship and you want to know where the bottom is, you have a sonar. And the sonar sends a sound wave down, then it's bounced back once it gets to the bottom. Well, there's animals in what we refer to as the deep scattering layer that will give you a little bit of a false bottom. And it's primarily for animals, it's Ninomiya off our coast. That's just one of the main animals of the deep scattering layer off California coast. Here's another animal called the siphonophore that has two bells to it. So in that case, it's very much like its jellyfish. And it's got a stem that comes off, and look at the tentacles that are streaming down. This is a true jellyfish. So now we've gone to a group called the scyphozoans within the cnidarians. They have stinging cells like all good cnidarians do. This one is eating a myctophid, or a lanternfish that lights up. And the reason I wanted to put this close to to the bioluminescence is because notice the shroud that is inside the bell itself, very dark. It eats a lot of bioluminescent food. And if you were to have a clear stomach and you take in bioluminescent food, the animal that you're eating might indeed be flashing off and giving away where you are to your predator, something that would eat the jelly. So we believe that over time this has evolved, so that this animal has a nice dark shroud that keeps the bioluminescence from giving away the position of this jelly in the deep sea. These are animals called ctenophores, one of my favorite animals. They're called sea gooseberries, comb jellies, all of these things. They're in a phylum all by themselves. They are not in the cnidarians, even though they look very jelly-like. That's because although they have cells to catch food, they're not stinging cells, they're sticky cells. They're called coloblasts. Well, this particular type of ctenophore, it's called Burrow E, doesn't have tentacles. And they spend their whole life taking one end of their body and just unzipping it. It's almost like Velcro. It looks very much like mucousy Velcro. Opening its mouth, and it eats other ctenophores. Even smaller Burrow E, but mostly just other ctenophores. So these guys literally are kind of ocean-going vacuum cleaners. And when you get them, sometimes you can literally see the other ctenophores inside their gut. This is some of the ctenophores are some of the more beautiful animals you will ever see. However, so few people see them because, folks, these are almost the consistency of smoke. That's how delicate they are. When you see them, and I see them diving every once in a while or swimming, I can take my hand and make just a flow of water, and it will not break them up. I don't do it that strong, but I could if I really wanted to. It wouldn't take that long to break up some of these ctenophores. Another really interesting animal that you've probably not heard of is in a phylum all by itself too. These are called the arrowworms, or the chetaignaths. And chetaignaths, the biggest one in the deep sea, show me your pinky finger again. That's as long as the longest one is your pinky finger. And yet they're about the diameter of a matchstick, a wooden matchstick, but totally clear. So you won't even know that you're seeing them. This is what they look like with the light right behind them. And I also show this slide because they get to be very prolific. So these chetaignaths, arrowworms, are very important in the plankton, in the midwater and on the surface. And these animals are totally clear, and they get terribly abundant. They're a very important carnivore in the deep sea. The snails are well represented in the plankton, not on the bottom, but in the water column itself. Together they're called sea butterflies, as you can see right here. Some of them still have their shells that you can see right here. And this one has another shell that you can find. These are very often caught in the Neuston. And here's one that has lost its shell over evolutionary time. It's called corolla, like the car. And you can see the big foot has been modified into a wing. And you can literally see the cross-hatching nature of the muscle bands in the wing itself that enables it to swim through the water by literally just beating its wings. Some of them get really bizarre looking, as you can see here, totally clear, except for the gut, also referred to as the nucleus in this group of animals. And they have their retinal cells in their eyes. They're visual predators. They're also very active carnivores. They have, like every good snail, teeth on their tongue, which is called a radula. I have such an educated group. I'm so impressed by that. They have that. And here is another relative, and I refer to this group as Snuffleupagus because they have that nice long snout. This animal looks like it's upside down. This group of mollusks still has a little bit of a vestigial shell, a shell that's still kind of left there from evolutionary time. The gills won't even fit inside, and obviously the rest of the body does not fit inside that little shell. It's got huge eyes that it uses, and it lives what looks like upside down. The foot is modified into a fin that beats back and forth in the water, and that's how it moves. And this animal, again, an active predator. Now we get to the squid. Squid are always kind of great. They're in the mollusks also. They're also cephalopods. There's even relatives of the octopus found in the midwater. And notice that the body of this octopus pretty much clear. It lives in an area where there's no place to hide. It's not going to be able to hide in a rock. So being clear is a real advantage. Now this is called the vase squid. We have an example of that in our exhibit. This particular one, scientific name is Histioteuthis. And Histioteuthis is interesting. I'm going to step away from the mic so I'll talk a little louder. Because each one of these little buttons you can see here is actually a photophore. And even though it's a little hard to see, this eye is smaller than that eye. So it's got two eyes, and they're of unequal size, even in a naturally healthy animal. The animal lives suspended so that the big eye faces up in the water column. The little eye faces down. All around the little eye are photophores. The big eye is detecting the amount of twilight coming down where these guys live, and the little eye is adjusting the intensity of the photophores to match the downwelling light. Now you might say, come on, how do we know that? Well, there was a scientist that went out on a boat, got a hold of some of these Histioteuthis, they don't live very long by the way, put them into a lab right there on the boat, put them into a container, had a light coming from the top with a rheostat attached to it, and had a light detection device underneath. And as they changed the intensity of the light downwelling, they detected the exact intensity of the light being given off by those photophores. So if you were a predator and you were underneath this squid looking up, and if the light was coming down and the photophores weren't on, it would cast a shadow. It would be a shadow. But if you look up and the photophores are matching the downwelling light, it would obscure its downward shadow. So we refer to that as counter-illumination. So really a cool thing with these little squid. This is a squid that made quite worldwide attention a while ago from right here at the Cabrillo Marine Aquarium. Because this little guy called the piglet squid, and it doesn't look at all interesting here, does it? And you see a penny right there to give you an idea how big they are. It's called the piglet squid because there's pictures, you can look at it online. It kind of looks like a little pig the way it's suspended in the ocean. But this is the photo that our photographer got of the piglet squid. The minute we saw this photo, we knew we had something. We put it on social media many years ago, and it's not too many months that go by where we don't hear about some place in the world that has seen this photo. If you Google this, trust me, you will see plenty of examples. We have been approached by plenty of places in Japan where they want to get this animal live so they can see it. And yet it's all about the way that the picture was taken. So the mantle has been all filled up with water. The animal itself, when it's alive, is kind of clear. You can see its eyes. This is the siphon, not a nose. And these are the tentacles, not crazy scientist hair. So this is all thanks to Gary Florin. One of my favorite animals you find in the cephalopods in the deep ocean is called the vampire squid from hell. How can you not like that? And the scientific name, everyone, is Vampiroteuthis infernalis, which literally is translated as vampire squid from hell. They're black, they have two big, huge photophores on their body. We normally see them about this big, so about as big as a pear. I've seen them maybe twice that size occasionally, but they tend to be pretty small. Don't know much about them. The Monterey Bay Aquarium has been working very hard over the years to try to keep them alive for longer periods of time, to learn what they eat, to learn how they reproduce. We don't know anything about these animals virtually. Here's a copepod. This is for Dr. Julie Passarelli, but it's not Parasitic. That's what she likes, parasitic copepods. And this is also a huge copepod. Notice that the antennae right here span more than 2 centimeters. I mean, that's like a monster of a copepod. Okay, I know I'm not selling it very well, but it really is big for a copepod. They're much smaller than that. These animals keep that black coloration, and they have one photophore that's very, very bright. So when those photophores fire off, it really, you can see it, and it kind of glows for a little while. This is a group of shrimp-like animals called mysids. Their body is red. Notice the theme of the colors again. This one is probably about a little bigger than your index fingers, to let you know how big they are. This is interesting in that when it's startled, it has the ability to put out a bioluminescent cloud. So it actually spews out almost like an ink, like a squid, but it lights up. So that if you were a predator going after this animal and you were getting ready to take a bite, and suddenly there was a cloud of light in front of you, it would certainly startle you in a normally dark place. Krill. How could we not talk about the deep sea and talk about one of the more common animals we find? These are the euphausids, or the krill, and we find these in virtually all of our trips. And this is what enables whales to get so big. Those baleen whales are feeding on animals probably at the biggest, the size of your pinky finger. A little bigger than that are some of the true shrimp. This is Sergschts. Sergschts is interesting in that it's got that red coloration, it's got a clear body, but it also has very long antennae that in this particular one are hard to see, but they go out and they're hinged 3 times. And they're probably twice the size of the original shrimp. And what happens with this animal, what's been shown by video, is when an animal comes up and it hits one of those antennae, the Sergschts goes right over that area and grabs that meal. So it's almost like those antennae are like fishing poles, where it's detecting where the prey items are. These are amphipods, another group of crustaceans you find. The ones that I have by the arrows here are benthic, that means they're in the bottom. But these two right here are pelagic amphipods. So they're found in the water column itself. And the group, as a group, most of them are clear. So clear that you literally can't see them until you take them out of the water so that you can actually see them. This is an illustration of an animal that I did my graduate work on. This is called Farnima sedentaria. Everybody say that with me. Ready? Farnima sedentaria. Very good. That literally translates as clever sitter. And it's also given the common name Cooper of the sea. And that's because the females make these barrels that are clear. So the structure is actually a barrel. It's made up of salps, and I'll show you pictures of those in a minute. And that was one of the things I was interested in studying. I also thought it was interesting that it was called the Cooper of the sea. Many of you may know that coopers are people that make barrels. And coopers are particularly famous in the old whaling days. And they're all men, by the way. Women were not allowed out on whaling trips. It was only men. And they would go out for years at a time, and they would take not empty barrels on the boats, because they'd be out for so long a time. They would bring planks of wood. And as they captured their whales and rendered the whales down to make the oil that they brought back, they would make the barrels and put the oil inside the barrels they were making. And the names of the title of the person making the barrels were called coopers. I always thought it was interesting that in Farnima sedentaria, the most famous of all coopers of the sea, only the females make barrels. Well, back in those days, like I said, no females were allowed to do that. This is a picture from Discover magazine just a couple months ago. I really liked it because it shows Farnima kind of half hanging out. This is all eye, by the way. You've got the mouth parts right there, and then there's a lateral eye and a medial eye and a lateral eye. So think of having eyes right here that face that way, and 2 little eyes by your jowls. And we think that the reason that they have these eyes with different retinal distances is the jowl eyes are designed to be able to focus inside the barrel, whereas these eyes are focused outside the barrel so they can grab food. They're active carnivores. They also, the females, will keep eggs in her marsupium, which all amphipods are known as. They keep their eggs there that are fertilized, and they hatch as little tiny amphipods like adults. And then once they hatch, the female takes them out with these big claws, gnathopods, and inserts them into the inside of the barrel, tail first. The babies eat the barrel so that the barrel over a short amount of time eventually disintegrates. Now, for my master's thesis, I was interested in knowing, are they really eating the barrel, or is the barrel just deteriorating naturally? So one of the things that I did was I knew that the outside of this barrel, made from a tunicate, a salp, or a pyrosome, that substance is called tunicin. And tunicin is very closely related in chemical composition to cellulose. So all I did was I did a simple test for cellulose, such that when I put the substance in there, it would turn a dark blue. And I did it, and indeed the guts of the babies turned dark blue. So it was kind of a proof of theory that these guys are indeed eating the barrel as they grow up. And eventually they will swim away to grow their own barrels, or to make their own barrels. Well, thank you. Farnima is so proud. These are worms that are found in the open ocean. And worms, I think most of us think of them as benthic, as bottom living. And yet there's a whole group that live in the midwater itself. And this particular group of Tomopterus, as it's called, is pretty big. Tomopterus gets to be 8 or 9 inches in length. This particular one has died quite some time ago when we took the picture, because in life they are totally clear. You cannot see them. There is a color morph that we've been interested in studying. We haven't seen it very much, where the gut is purple. And that is very interesting. And I don't even know if the literature has dealt much with this particular color morph, but something that would be interesting to study. And you may have noticed that many of the things I'm telling you, I tell you that we just don't know the answers to many of these questions. We finally get to our own phylum, Chordata. And these are tunicates. They're still invertebrates. They're not in the vertebrata. They're invertebrates. But these are tunicates, and these are ocean-going sea squirts relatives. This particular group are individual salps, as they're called, whereas this one down here is a colony where each one of those little triangular structures you see is an individual zooid, or an individual salp-like animal. And they're a different group called a pyrosome. So what Farnima would do, to get back and finish that story, is she will crawl into one end. She'll grab a hold of this, crawl into one end, eat the inside muscle bands out that are lining the inside cavity of the salp. Then she will cut it off to be the right length for what she needs to make her barrel. She will shape the inside so that it's bigger diameter than either end, and it'll be just like a barrel. And that's what she lives in. And she will then have her brood, put her brood on the inside of the salp, and then the babies will eat it. She does the same thing for this pyrosome. She crawls into one end because it's open on that end, closed on that end. This is a colony. They get to be anywhere from the length of your finger to, if you can imagine the top of the colony be the top of the roof, all the way down to the bottom. 10, 15 meters in length. That's how long they can be. They're called pyrosomes, which means fire body. They're brilliantly bioluminescent, by the way. If you ever get a pyrosome, it'll really light up if you put it in a dark room and it's still alive. So to finish the story, the female Farnima will crawl inside. She will cut it off. She will, with her claws, grab individual zooids and pull them out. And what was interesting was, when I was studying this, I thought, well, shouldn't some of the barrels have holes in them then? Because that's where the zooids used to be. But what we noticed was, when the female Farnima pulled the zooid out, it closed up so that it became solid again. So that the barrels of a pyrosome, although look different than the salp, they're still no holes in it. So it was kind of an interesting little conundrum that we had to solve when we did this work. This is one of the more common salps that Farnima uses for food. It's called iasis. It's probably about the length of your index finger. You're starting to get the idea that these are small, I hope. And it will eat this nucleus, Farnima will, and then cut it off, eat the inside, and use it as a barrel. Shape it and use it as a barrel. These animals go through life by taking water in. The water goes over a gill bar. The gill bar has mucus on it that is constantly being replenished and brought towards the mouth. So as the tiny little plankton get caught in the mucus, it eats the mucus, and that's how it feeds. These are tunicates that never grow up. So you can think of these as Peter Pan tunicates. Because tunicates, sea squirts, they start out life as the tadpole stage when they're very small, right after the fertilized egg. And then eventually they will land on a boat dock, and trunk first, they will connect to the boat dock. The tail will go away and it'll grow into a sea squirt. These particular types of tunicates, called larvacean tunicates, they never grow up, they're larvae. They keep their trunk, they keep their tail throughout their lives. They also are very interesting in that they make a mucus house. This mucus house is made by the trunk of the body. This is the tail that goes back there. This right here is a mucus netting made by the trunk of the tunicate. This is a feeding structure that has mucus on it, so that when the water comes through the house right there, only the smallest particles are allowed through the netting. Then what it does is the smallest particles get caught on the mucus of the feeding structure, and then the mucus is eaten by the tunicate body. The tail beats rhythmically, moving water from there all the way through the house to the outside. And when it becomes clogged, which happens quite a bit, the animal just flies right out of that little trap door right there. So it's got a trap door that it goes out of. And the house just settles down to the bottom, filled with all that good stuff that's still stuck on the outside. And this animal has a remnant of a house right on top of its trunk. And as it beats its tail and wiggles back and forth, it's inflating the new house that it then flips into, and this goes on all over again. So you can see some of this plankton really has very unusual life histories. To make it even more interesting, at least to me, there are little granules that are embedded in the walls of the mucus house that are brilliantly bioluminescent. So bioluminescent that there are some bays and harbors of the world where most of the bioluminescence that we see is the result of these larvacean tunicates. The house is about the size of the tip of your thumb, by the way. And they can be much smaller than that, the tip of your finger. So that's how big they are. This particular one, by the way, is a deep-sea larvacean tunicate called Bathycordius. And Bathycordius makes a house, but it's like a big umbrella in the open ocean and the deep sea. Very interesting animal. We finally get to some vertebrates. Here's the fish that you find. We have animals like lanternfish in the groups called myctophids. They have little photophores all on the outside of their bodies that light up. They use that light to find food. They use that light to find mates. It's a great way to do it. Here's an animal called a hatchet fish, because it looks like a hatchet. And it has photophores that are directed down so that it can counter-illuminate its shadow. It's found in the midwater. Here is a deep-sea angler fish, and that fishing pole right there terminates in a lure that lights up. And then here, folks, and I know this is going to be a hard sell. Everybody show me your pinky finger. Pinky finger, pinky finger. They're half that size. That's how long they are. And they're very thin, and they are the most common vertebrate in the world. And we say that because they're found in all oceans at many different ocean depths, such that we believe them to be the most common vertebrate in the world, Cyclothone. Some of these fish get pretty bizarre. Here's a gulper, and it has a jaw much like a snake, that it can unhinge and eat a bigger fish. It has a huge distensible stomach that it can take a very large prey item all at one time, but it doesn't have to eat all the time, which I think is what makes that good. And then here is a black-bellied dragonfish. We get those every once in a while, and I'm happy to tell you, for a change, these get big, at least as big as the midwater does. They're about as big as my forearm. I've seen them almost that big, and their diameter is about like that. So they get pretty good size. They also have very impressive looking teeth. They have photophores lining the bottom part of their body and all over their body that can light up. And they have photophores on either side underneath the eye that look like red patches. And when they bioluminesce, it's red bioluminescence. Now, I've told you, at least if I didn't, I want to make it clear to you that we don't know near what we want to know about ocean animals in the deep ocean. But here's an example of a paper that came out in 2006. You all know about DNA sequencing. It's part of our life, part of our world. Well, here is an example where some of those sea butterflies were actually being sequenced out at sea. So they were getting these animals and sequencing them. That way they could start understanding how these animals are related. I also wanted to remind you that the ocean is a big area that absorbs carbon dioxide. Primarily the carbon dioxide we are spewing in from our burning fossil fuel activities. And we all are responsible for that. Well, the open ocean has been absorbing that, but studies show now that it's absorbing about 10% less of the carbon dioxide than it did even 100 years ago. Now that's concerning, because now we already have a system where there's a lot more carbon dioxide going out, some of that carbon dioxide staying in the atmosphere, causing our atmosphere to be kind of like a heat-trapping blanket on the outside of the planet. We're warming up. We have plenty of studies that show that. Well, if the open ocean starts losing its capacity to take carbon dioxide in, that means we've got to take that into account as we come up with the different scenarios of how we're going to adapt to a changing climate over the next 50 or 100 years. Sea butterflies are, boy, there's a theme with sea butterflies. They're referred to in this particular paper as canaries of the sea because they make a hard outside shell, and it enables a certain, what enables it to have that happen is a certain pH. And pH is a scale from 0, very acidic, to 14, very basic. And living systems tend to be at about 8 on the pH scale, 7 being in the middle. So 8 is about right for a lot of living systems, and ocean water is best about 8.1 or 8.2. Well, these animals are adapted to make their shell out of the calcium that's in the water by having a certain acidity that allows them to do that. If they don't have that, if it's too acidic, even like 7.9, 8.0, they start showing the effects of not being able to make that shell so reliably. Corals are another famous one for that. Some of this has been done right here on the West Coast. This is a paper that included this data, and what I wanted to point out to you is that here is a data set that shows what the surface water pH was about 1750. This is 2005, and this is what's projected for 2050 on one of the scenarios. Just to remind you, there's 8.1 in the blue. Once we get down into the green, it gets into 8.0. And once it gets into the yellow, we're talking about something even below an 8.0. That, even though it doesn't sound like much, is enough to cause some real changes in our local ocean and for the organisms that live there. So we've got to keep these things in mind as we make the scenarios for what we're going to be adjusting to. Another thing that I like to end my kind of talks with is, where did I come from? Here's Dr. Charles Gault, who was my professor at Long Beach State. He's now retired and living up north. I still get a chance to keep in contact with him. You can see a young Mike there. And then this one in the middle is Dr. Lisa Gershwin. At this point in her career, she was Lisa Gershwin, and she was studying jellyfish here at the aquarium. She became very interested in that. And I refer to her now as the jellyfish goddess. She has identified virtually dozens and dozens of jellyfish new to science. She finished her PhD at James Cook University in Australia, where she has become the marine stinger advisor for the whole country of Australia. And they call their jellyfish marine stingers. And they're very interested and pay a lot of attention to jellyfish in Australia because they kill people regularly every year. And we don't know enough about them to help that from happening. You can see Lisa looking at a box jelly that's found off Australia here. She was also part of an active research project that had the story right here at the Cabrillo Marine Aquarium. This is called the black sea nettle. It has shown up in the 19th century, or in the 20th century, in the 1900s, only 4 times in Southern California. 4 times. So we didn't know much about it. By the way, you'd think it must be pretty small if it's been overlooked all this time. No. The bell diameter is 1 meter. It gets up to 3 feet in diameter and literally probably 10 meters long with the oral arms and tentacles that hang down. It's a huge animal. And this one is a young one that we grew here at the aquarium. When they get bigger, they're almost black, which is how they get their common name, black sea nettle. It's actually a very dark maroon color. But Lisa was involved because it didn't have a scientific name. Lisa became part of a team that gave this name, this animal the name Chrysora achleos. And how it becomes interesting here at the aquarium is we have one of the early specimens that was used as the type specimen for this jellyfish. What that means is that if anybody wants to study this jellyfish and they want to know what it looks like from what it was named from, they have to come to the collection at the Cabrillo Marine Aquarium in order to do that. Now, we don't have a lot of samples like that. We have a couple here and there. But this jellyfish is one of our most precious in our collection that we have here at the aquarium. Lisa also wrote a book recently called Stung, and it's all about how jellyfish in a changing ocean are going to adjust to it better than other animals, such that we expect to see the jellyfish blooms that are more and more common to continue into the future as our seawater changes. And a bloom of jellyfish may make people like me pretty happy. I like seeing jellyfish. But we use seawater to cool local power plants. And there has been examples all over the world where cooling water, the jellyfish are so abundant, they come in with the cooling water and they close down the power plant. They literally close it down. They get into intakes of ships, and they can ruin the engines of a ship that would use the seawater to cool as a radiator. They also just fill up nets of people that are fishing, and they get almost all jellyfish and very few fish, and so much so that they break the nets. So jellyfish blooms are a big deal, and there are regular symposia trying to understand more about them. But one thing clear, it appears that we're going to be seeing this more often. By the way, Lisa also has done a TED Talk on this, and she has been, she's just about ready to publish Stung 2. Stung 2 coming soon to you. So the end of my talk was that nice view of over the ocean, because I want to keep a serene feel to the open ocean, because that's what it's like. Thank you very much, everyone, and I'm happy to take some questions. We have time for a couple questions. Yes?
Why is the ocean absorbing less of the CO2?
Why is the ocean absorbing less of the CO2? One of the obvious reasons is as it absorbs it, it's not being used as quickly when it's in balance, and so it's kind of filling up, so the capacity of the absorption is actually being affected. But that still is being actively studied. I think there's more to that story. Yes?
So when you say the water goes, as you go deeper into the water, is the current a lot less that they can survive down there versus up in waves and stuff like that?
There is a current so the question is, as you go deeper, is the current stronger or not as strong? And the generic question, or the generic answer is that it tends to be less energetic as you go deeper, but it's a much more massive flow. So when you get to the bottom of the ocean, there's water that makes hundreds of years traveling to finally get back up to the surface waters. So yes, but it's very, very slow. So in the deep ocean, you don't find the currents you find in the shallow continental shelf areas.
I mean, because they just sound so fragile. Like
They are. And when you see them from the submersibles, there's not a lot of movement going on. You're going to have to take a lot of video and fast forward. Yes?
You said that there's more jellyfish bloom recently going on, but it seems that this past year, I only saw one jellyfish swimming every day. So where were they?
Well, I guess the best way to say that is this is kind of on a regional basis. So where are the jellyfish if there are really more jellyfish blooms? That particular comment is taken more on a planet-wide view, so that in certain areas, like fjords in Norway, off Japan, there's a huge jellyfish called Nomura's jellyfish. If you've seen pictures of one that looks like the size of a Volkswagen, that's Nomura's jellyfish. And by the way, they aren't that big. That's because the perception has been stricken quite a bit. But the point is that they are still really big, and they're causing all kinds of trouble for the fishermen there. However, off our coast, when I was growing up, I grew up in this area, and I would go body surfing. That was one of my favorite things to do every single summer. And there was jellyfish season. That was what we knew we were going to get stung. And if you looked around enough, you'd be able to see what's left of a purple striped jelly, that you were getting stung by a piece of it. So, but I don't see that so much anymore. And we don't know the answer to that. Not all jellyfish will be winners, if there is such a way to put that, in the climate change issues. Yes?
You mentioned something earlier about deep scattering layer. How like, sometimes we got people got confused with it being like a big bottom. Like, what was it you said earlier again? Like, what was it that big top?
So the question of the deep scattering layer is, what did people think it was? I may have oversold that a little bit, because when sonar came around, and basically it's sound waves that are being sent down, and then when it's being reflected back up, it's being detected by the sonar itself. And this is very interesting for, I think all of us know this from war movies where they're trying to find out where submarines are and things like that. So when the sound waves are putting out, by the way, marine mammals use that to catch their food. They put out these sound waves and they bounce back. But in the case of what I'm referring to, the deep scattering layer, this is from a boat at the surface, and this is the bottom of the ocean, thousands of feet deep. And when the sonar sound is directed down, it will eventually bounce off the bottom and come back up and let you know the depth. So you know exactly what that depth is. However, these particular animals that I talked about, they actually, when the sound hits them, the sound is reflected off their bodies, just not all of it. And so you end up with what looks like a ghost shadow, otherwise known as the deep scattering layer. And you can actually follow the deep scattering layer to see diurnal migration, where you'll see animals in the middle of the day the deep scattering layer being very deep. And then as the day gets older and eventually into night, the deep scattering layer can be very close to the surface. Any other questions? Yes?
Aside from maybe extracting proteins or compounds from jellies for medical research, is there any other widely commercial use for zooplankton in general?
Well, there's been a lot of work. So the question is, what kind of benefit can we get from zooplankton if we collect it? First of all, it's very expensive to collect, so the business model may not always work out real well. I do know that the Russians many decades ago were collecting a lot of krill, and they were taking the krill and making it into a paste, and it was very high in highly unsaturated fatty acids, hufa, and they figured that would be a great protein source and lipid source for people eating it. The problem is that it was so rich in oil that most people, unless your gut was rather adjusted to that, it would give you terrible indigestion. So it didn't work out real well from a commercial point of view. But jellyfish are, in Asia, a very common, they're added to salads and to other food types. There are people that have contacted us over the years that have asked us to How do we grow jellyfish? Because they know we do it, and they want to do it commercially so they can sell the jellyfish. I still say that that's a difficult business model, I bet, to keep going. Maybe one more question in the back, and then we'll...
Mike, you mentioned that some of the creatures undergo a vertical excursion of several thousand feet.
Right.
For example, sperm whale is one of the amazing, but how do these creatures that you showed us, how do they accommodate the change in pressure? How do they exhale or inhale to accommodate that change in depth?
Right, so great question is, how do these animals that take this thousands of feet deep, I gave you the example that when you go down to the bottom of your swimming pool, your ears hurt unless you equalize. I want you to imagine to be in one of the deepest parts of the ocean where the pressure is so great, it would be the equivalent of having a school bus balanced on your head. That's the pressure that you find in those areas. And not only on your head, it would be school bus on both sides, everywhere of your body. We are not adjusted to do that. In the case of sperm whales, I don't know a lot about this, but I'm going to look to Diane and Bernardo to help me. But as they go down, their air passages just get shrunk. Just get squeezed, because that's compressible. But the rest of their bodies, for the most part, not so compressible with fluids. The animals that I was referring to, for the most part, most mesopelagic animals, most deep water animals, do not have air passages that are compressible. So they don't have those kinds of problems. What I've always thought curious, and I don't think I've ever seen this studied before, is that I'll bet metabolic pathways are affected by pressure. So that digestion, reproduction, all of these actions that happen in the deep sea, and then you bring them up to our type of less pressure, I'm thinking some of those metabolic pathways may not work.